right now. We are very fortunate that Don and Peg Krisst have joined Beit Knesset Emek Rafaim and that he can speak with us here today. I wanted to introduce Don with something about his remarkable medical achievements, places like Johns Hopkins, Stanford, University of Maryland, Rabin Medical Center, which you can read about in the web post announcing this talk. So I asked my wife, Diane, for help, and she said it much more plainly than I ever could, and I think it's a good introduction. Professor Don Krisst is a smart guy, and he knows a lot about epidemics and viruses, so we should all listen very carefully to him. Don, take it away.
Okay, thank you very much. I will say, I think that this will be the safest exposure to corona any of us will ever have. And hopefully it will be a didactically beneficial one as well. Okay, hopefully everyone can see my shared screen. I'm going to try to make it in plain English. These are basic. Sorry. These are basic aspects of what you hear and read about continuously over the last several months. So my goal is to talk about the COVID-19 pandemic, which is the name of the disease, due to the SARS-CoV-2 virus, which is the name of the causal agent for the disease. Okay, and that my goal is demystification, which means not an encyclopedic treatment. It's a background for a period of discussion to follow. As I've given this before, and that's always been the format. Now, what I'm going to focus on are some of the numbers you hear about and people talk about. That's called epidemiology. Then we're going to talk briefly about the biology. That comes in two phases, the biology of the virus itself, and of course, the clinical aspects, the human biology, the reaction to the virus. And based on those two considerations, we're going to advance into prospects for cure. And I've added more or less a semi-philosophic medical synthesis at the end about the way forward, given everything I've talked about up to that point. So let's start with the epidemiology, the numbers that you hear. So we're in a viral epidemic that has become a pandemic throughout the world, and the COVID, the virus, the SARS-CoV-2 virus, it was called novel coronavirus for a number of reasons. And one of the reasons was that it was incredibly highly transmissible. In my experience with multiple epidemics in different parts of America and different characteristics of the epidemics and different kinds of diseases, I've never seen a disease as transmissible as this. I mean, it's almost a handshake and a sneeze and you have the virus. And that means, in terms of translating that into numbers and to an image, you have something called an exponential curve. And that's what everyone is talking about. The exponential curve records or documents a rate of change. And actually, it's the change of the rate of change that you're seeing. So it tends to have this explosive course rather than a straight line. And it can come in different degrees, different slopes, we say. A very mildly progressive epidemic would have more or less a flatter slope. A fairly somewhat more aggressive would have a slightly higher slope. And then you have this, which is fairly aggressive, but this is sort of the case of the coronavirus. It's extremely, extremely transmissible from one person to another. Now, what's the basis for this mathematical expression? What happens in the population among people? So you start off with a few individuals, and because it's highly transmissible, you have the people, the contacts of these individuals, which are now at extremely high risk. And what happens after a very short time, you have the index case, and he has infected all these other people. However, there are still lots of people that remain to be infected. So every one of these then becomes a messenger of the virus to the rest of the population. Okay, now what everyone is talking about, and it gives hope to epidemiologists and specialists and experts, is flattening of the curve. So why do curves flatten? Well, it's an exponential curve. And practically everything in nature, physical phenomenon, biological phenomenon, follow this sort of curve, that they grow exponentially. And at some point, they begin to flatten. They reach an inflection point, and they make a decision that now they're not going to behave the way they have been. They're going to behave more mildly, and they flatten out. They flatten out for a very good reason. In the case of a viral epidemic, they've exhausted all the possible reservoirs of virus, people that have the virus, and people that have had the virus. So there's no one to give to be a source of the virus, and there's no one to give the virus to. So the epidemic flattens out and becomes benign. Bacteria in culture do the same thing. They've exhausted the food they survive on, the substrate in the petri dish, and they stop growing. But in the initial phases, they grow like this. Now, everyone is worried and hears much about, in Israel, a second peak. So I downloaded this bit of statistical information from about 2-3 days ago, which meant it was 4 or 5 days old. And you can see this is the way it was before, say, May 27th. And it was behaving exactly like one would expect. It was growing exponentially, with a very slow start here. And then it exploded, but then it reached an inflection point. It was beginning to flatten out. And now there is a slight uptick in the incidence of infections. And this here documents in a more specific way. You get a feeling of actual numbers compared to what it was in the peak. It's growing in a progressive way, but nothing quite as scary as it was way back in the beginning of April. So what the future of this is, we'll have to watch it very closely. I have some global comments about how we should be thinking about it, which I'll address at the end. Okay, now that's the epidemiology. Let's begin to talk about biology. I want to start first with human biology. And the key thing that is near and dear to all of us is the symptomatology. How are we going to present if chas v'chalila we actually contract? People that contract the virus, what happens to them? Well, first, they have a cough. And of course, frequently, you would expect them to have a pneumonia. Bacterial pneumonia is what we call productive. You have a cough which produces mucus, phlegm, and so forth for various reasons. The amount of fluid in the lung with a bacterial pneumonia, it tends to be rather large, and so you have a productive cough, a cough that produces these kinds of fluids that can be sampled or whatever. In particularly many viruses, but particularly in corona, it's striking that the cough is very dry. And nothing comes up. There's nothing, not a lot of fluid downstairs in the respiratory system in the lungs itself. So you have a dry cough, and then you have very interesting changes in taste and smell. Now the data is only about 3 weeks old, and it came from, interestingly enough, an application, a popular application that was distributed all over the world, and it had millions of users. And the idea was, if people had something they thought was a corona infection, they'd record the symptoms, the presentations that they had. So little by little, there were millions of documented cases. And what came out of this was that there was a surprisingly high percentage of people, a third of all the people that responded, had changes in taste and smell. That immediately set off a little alarm in my head. I said, hey, that's behaving like some sort of inflammation of the meninges, a meningitis, something along that line. And a meningoencephalitis, a low-grade one, is not uncommon in viral infections in general. Ordinary flu and a number of other viruses will give you a full head sometimes, a little ringing in the ears. That's an expression of this very low-grade meningoencephalitis. So the fact that it's focused on taste and smell, which is very interesting, these are controlled, the sensory aspect of it, by nerves from the brain itself, as opposed to the spinal cord, which obviously sends out sensory nerves to the periphery. But there are some nerves, there are 12 of them actually, that come from the brain itself. They're called cranial nerves. Now, when you say taste and smell, that suggests some specific cranial nerves that are involved in mediated the sensory information and transmitting it back to the central nervous system. So we have specific cranial nerves, and we have, again, everyone knows a specific cranial nerve for smell located in the nose, the olfactory nerve. So you have some involvement, probably inflammatory involvement of these nerves by this very low-grade meningoencephalitis. This is going to become important later. Both of these nerves service the face, mucous membranes of the face, the mouth, the nose, and everyone talks about the eyes. So these are things that you should keep in mind at this point, and we'll return to at the end when we think about what we're going to do with our lives. Now, the other thing that comes up very logically for a virus that's supposed to be transmitted by droplet infection, that's a complicated issue, whether in fact it is or it's the main avenue infection isn't resolved. Opinions and data change from day to day. But let's say for the sake of argument at this point, that's likely the case, that we wind up with a pneumonia. And so you're going to have difficulty breathing. And this is what the chest x-ray from a person with a typical viral pneumonia would look like. You have this upper lobe region, which the metaphor is ground glass. If you ever seen ground glass, it's used as an optical filter to diffuse light. This is normal lung. It's black. It's radiolucent. There's nothing to prevent the x-rays from going by. And what it represents is basically an air-filled tiny little sac. And there are hundreds of thousands, maybe millions of them within the lung. And that's where this is a blood vessel surrounding these little sacs. And that's where the transport of air occurs. You take an air from the atmosphere and it's transported through the rest of the body, through the lungs, and that transport occurs at the level of the alveolus. Here, the alveolus, there's something wrong with it, whether it's filled with fluid or whether it's damaged another way, but it doesn't look like a very healthy lung. It doesn't look like air transport or oxygen transport is going to be very efficient. So you have difficulty breathing. Now, we'll return. There is another very interesting and important aspect of that I'll go into in a second. The second thing, third thing, which is fourth thing actually, which is very interesting, is that there is a low-grade fever, but it's a little higher than what you normally see with viral infections. Most viral fevers are so low. There had been, when I was training, a real dialogue in the literature whether it's worth treating a viral fever, a virally produced fever, with antipyretics like aspirin, Tylenol, and so forth. Because you would, in order to lower the temperature, you would diaphoresis, you would sweat, and that's uncomfortable. So maybe it's worth having the low-grade fever for a few days, and it'll resolve itself, and don't take anything for it. Anyway, it was discussion. This is a little higher than that. People really know that they're febrile, which is unusual for a virus, and that's one of the reasons why people have these remote guns in airports and all sorts of places to see if people presenting have a fever. The other symptom, which is somewhat unique to corona. I don't know that it doesn't happen in another virus. I've never come across it in that context, but it's called silent hypoxia. Now, hypoxia is the lack of oxygen in your periphery. You're not getting enough oxygen. You would think that would occur predominantly with a scenario like this. And yes, in this kind of scenario, you have hypoxia and person without oxygen huffing and puffing, difficult getting enough oxygen in. These cases with silent hypoxia, and it occurs a significant number of cases in Israel. The Israeli cases that they've seen at Shaare Zedek, I know, often present with silent hypoxia. So normally your peripheral oxygen saturation, as it's called, should be about 95, 96, 98%. That's normal. When it gets to 90, 80, that's really seriously low. It can go lower than that, and I've seen cases like that in people who are chronic smokers and they have chronic lung disease and they have a chronic problem exchanging oxygen and taking it in. Those people can have extraordinarily low levels of oxygen in the peripheral blood. But in COVID-19, excuse me, you can have these very low levels and be asymptomatic. It doesn't bother you at all. And just incidentally, you may have a mild fever, you go to your doctor, and he tests you with something called an oximeter, I'll talk about in a second. He says, holy Moses, you have a PCO2, a PO2 of maybe 89, 85 or something like that. Anything below 94 is significant clinically. So he would hospitalize you. You have a major problem, or he might at least. That is often said to be the prudent thing to do. Every physician makes his own decision depending on the person, the setting, and other clinical factors. Anyway, that's called silent hypoxia. What is recommended for that? And it's a good alert. You can obsess about it. You don't need to. Perhaps twice a day. You don't always realize that you are getting sick. You use what's called a pulse oximeter. In fact, I have one. Looks like this. They're relatively inexpensive. You can get them from eBay. You can get them locally from other sources in Israel. It's not a big investment, and it tells you these two things: the level of oxygenation. It's not really lung oxygenation, oxygenation of peripheral blood. In this example, it's 98, which is perfectly normal. And as an internal control, it also tells you the heart rate, which is an indication that the device is working. It's battery powered. So if that's normal, then you can think that the value is normal. There are other sort of subjective tests that have been recommended. I find them, they're not quantitative and they're subject to hysterical interpretation. So I like quantification, and it's an inexpensive way of doing it. And it could be a heads up. It actually could save your life. I don't think that's an over exaggeration, especially for people who are somewhat vulnerable. Now, that's the human biology aspect. To speak intelligently about the options for therapy, I think we have to, just a second, let me put some light down. I see my camera struggling to correct the ambient illumination. Okay, I think that's better. To intelligently approach the issue of developing therapies or utilizing existing therapies for COVID-19, I think we really have to understand the basic biology of the virus. And people are scampering around the world intensely and vigorously pursuing that thought as well. And there is a type of electron microscope called a scanning electron microscope. And actually, the virus is large enough so that you can see it in that format. And the scanning electron microscope has the quality, probably unique. It was the first of the sort. There are other types of scanning electron microscopes now, where you can visualize these tiny structures in 3 dimensions. And what you see is you basically have a sphere, and off the surface of the sphere, you have these projections. And in someone's imagination, they reminded him of the peaks or spikes in a crown, and hence, which is a corona, and hence the whole class of virus was known as a coronavirus. Now, the SARS-CoV-2 is not the only virus of this family. Coronavirus actually has 7 key members. 4 of them are regular cold viruses, and they occur in most of the Western countries, including America, and I'm sure Israel. And they're benign. They're regular populations of our environment, and we get them from time to time in the winter as just common colds. However, three of the viruses may be associated with serious or fatal outcomes. One was MERS, which is the Middle East Respiratory Syndrome virus, and one was SARS, the severe acute respiratory syndrome virus, and the other one is our SARS-CoV-2. Now, the interesting thing is, which really has biological consequences and therapeutic consequences, this virus, our virus, the SARS-CoV-2 virus, contains the largest amount of RNA. It's the largest RNA virus, a way of putting it. Now, this is a schematic of this beautiful picture on the left. And the schematic indicates that the different basic parts of a virus. You have this nucleic acid, the RNA, is inside in the core, and surrounding it, an envelope, is a series of proteins and lipid. The outside is a lipid membrane, and you have a series of proteins that enwrap the coil of RNA. And moving from the core to the external surface are these peculiar projections, and they're all proteins. And some of them actually are fairly important. This one particular, which is called the spike protein, because it's sort of like a spike, right? Coming out from one person's imagination. He didn't see a crown so much as spikes. But anyway, they call this the spike protein. And its importance, as we will see momentarily, is that this is the way the virus gets into a cell in a host organism, not necessarily a person. This occurs in nature. This is the way this virus gets into a cell. We'll see why that's important. It obviously is important from one point of view because it mediates the infection. That's how people become infected. And this is a key protein in the process. The other significant thing to note is because this is the largest RNA virus known, it also, RNA is the thing, the template that is responsible for the production of proteins. So this virus produces more proteins than any other comparable virus. It produced 29 proteins. Now, there is good news and bad news. The bad news is that you have so many proteins, it has a lot of functionality. And so it's going to be a very mean target to overcome. The good news is that by having 29 proteins, there indeed are a lot of potential therapeutic proteins. There's this envelope protein associated with the envelope. There's the spike protein we've talked about. There's the other little proteins on the membrane, one called the membrane protein appropriately. And then the protein that enwraps the RNA itself is called the nucleocapsid protein. So all these proteins are legitimate targets for therapy. Okay, now one other thing. There is a very important general principle before I go into the details of this slide. This slide essentially justifies concern for the coronavirus and for this epidemic, and why it's not only it's highly transmissible, but why it turns out to be so fatal. Viruses lack an ability to self-replicate. So they are obligate parasites. They have to invade some host cell and cannibalize, pirate the metabolic machinery of the host cell to make new viruses and continue their life cycle and their deadly life cycle. And in the process of doing that, several things happen, which we'll talk about in a second. But let's talk about, we talked about the spike protein, and that was important to the entry into the cell. So now you see why it's important to get into a cell. It's the way they replicate. If they don't replicate, you can have a huge amount of active virus on the surface. Eventually, the virus will break down into its component chemicals that you've seen before, and it becomes non-infective at all. You could lick the surface after a while if you choose to. It really is of no concern because the virus then becomes non-infective. In order to remain infective, it has to self-perpetuate itself. It has to be able to reproduce. And that's what this very clever mechanism ensures that it can do. And it does it, unfortunately, at the host expense, whether it's a bat, whether it's a human being or some other animal. That is the way the Ribono Shalom set up the world, that these viruses function as intracellular parasites in the worst possible way, because they frequently will destroy the hand that feeds them. Okay, now, the way a virus enters, we know from the virus point of view, it uses a spike protein in some way, and we'll talk about that in a second. But for the present, I want to introduce another concept, and that's a receptor. There are proteins not only on the viral surface, on the viral membrane, but there are lots and lots of proteins on our cell membranes. Now, those proteins do various things. Some of them are actually receptor proteins, and the word is more or less intuitive. Excuse me. They receive chemicals from without the cell, like hormones, modulating substance, drugs that you maintain, that are going to act on the cell, right? If you're taking thyroxine, it has to get into the appropriate thyroid cell. How does it do that? It does it through a very clever scheme. There are receptors on the membrane of the thyroid cell, for instance, and a molecule of thyroxine is floating along the blood, and it sees the receptor, binds to the receptor. There's some sort of chemical reaction, and then that changes something about the receptor, so that the receptor then at the other end that's in the cell tells the rest of the cell, hey, now let's increase metabolism, or hey, let's do X, Y, and Z to make this person healthier. That's the way it works. It's a receptor-mediated process, or receptor ligand binding, and it's called signal transduction, if you like big words. And it means there's a signal outside the cell that gets information into the cell. All right. Now, that's the way it works normally. And it works very well. You control your blood pressure, your heart rate, and a whole bunch of things. Your brain works that way. Your gut works that way. The virus takes over that mechanism. And it does it because the SARS virus is enabled to do that because these spike proteins that we spoke about happen to fit perfectly into the human ACA receptor. ACA is one of the kidney proteins that regulate blood pressure, aldosterone, and this is an important function. There's the various ligands, again, float in the blood, and they find this ACE2 receptor and they bind to it, and they transmit information about what the cell may be. The wall of a blood vessel, a smooth muscle cell in the wall of blood vessels, so it's a contract, relax, whatever it does. Okay, those are the signals. That's how it gets there. Now, the COVID virus takes that mechanism over completely. It binds strongly, and that's one of the important issues here. It just, some things could bind very loosely. It's like playing catch. Someone throws you a ball, sometimes the ball just bounces away. If you're a professional ball player and someone throws you a ball, even if it's lightly, you catch it and you know how to keep it. Well, the virus, the SARS virus, knows how to bind very strongly. It has its molecular configuration of this S protein is such that it knows exactly the kind of things that this AC2 receptor is looking for. Now, it turns out this ACAC receptor, when it sees a spike protein requires another protein, as it's called a primer, a helper protein. We don't know what it does normally, but we know it in some way it's complementary as required for effective viral binding. Now, this is one way that the virus begins to enter the cell. Another way, you remember the virus is covered, enveloped by a lipid membrane. So that lipid membrane has the capability of fusing with your cell's lipid membrane, and then it forms a little vesicle out here. And then the RNA inside the virus is extruded into your cell. Okay, and so this is sort of this was a microscopic view. This is a macroscopic view of the process with an entire cell, so to speak. And here's the virus, and here are these little S proteins, the spikes, and they're engaging the membrane. And the membrane here is trying to envelop it, and it's going to enclose it entirely in a little intracellular vesicle. It'll dissolve the outside membrane and release the RNA into the nucleus. Now, that's important because the RNA becomes these little small bits called mRNAs, and every mRNA is the final pathway to generate a specific protein. So that's the template for a bunch of proteins. The virus lacks that ability completely. It lacks the ability for RNA replication. It has a few enzymes which are critical for the process that contribute to it, which comes packaged in the deal, and you'll see are going to be targets for therapy. But by and large is dependent on nuclear factors, and then in the cytoplasm, which is this large area around the nucleus, it's dependent on various aspects of the normal metabolism and normal synthetic processing of proteins that occur in the host cell. And here is something called a Golgi. It's not important the name of it, but this is the final packaging where the virus forces the host cell to actually put all these little pieces of protein together into a composite virus. And then in this case, it extrudes the virus as an intact virion. The other scenario often happens. It doesn't have an extrusion process per se, but the virus continues to replicate. And finally, there's so many virions in one cell, the cell explodes, dies and explodes, allowing then the release of hundreds of thousands of new viral particles then to infect other cells or other people. Okay, now, by itself, that might not be so significant. We all get colds, and cold viruses infect our nasal mucosa. You get some death of cells, and you may have mucus with cloudy mucus with representing cell breakdown and so forth. But it's generally not a major deal, and it resolves within 2 or 3 days. Now, sometimes it doesn't even have to be in a setting of infection. Sometimes in late August, you're confronted with an antigen, and it is allergy season, and you sneeze and do all sorts of these things. Now, all this is part, and you have runny nose and whatever, this is all part of a normal immunologic response, an inflammatory response to the antigen, that the flowers, the pollens, or whatever is getting into your nasal passages. It's an exaggerated response, and that's the important point. Now, you're expecting, when you have this kind of viral replication in cells and death of cells, some sort of inflammatory response. That's the way our bodies protect ourselves normally. And those kinds of infections, which are self-limited because the inflammatory the ability of a certain population of cells to defend you is working perfectly, okay? So that's successful inflammation, and these are an array of cells. Now, one thing that happens peculiarly, I won't go into the details of it, but it becomes an overly intense, somewhat bizarre. These normally well-regulated inflammatory cells lose their regulatory compass, and they just produce huge amounts, excessive amounts of a kind of chemical called a cytokine, which is normally the way the cells arrive at a scene, secrete a cytokine, and the cytokine is helpful in controlling a foreign invader and infection, stuff like that. So cytokines are very important normally in controlling tumors, for instance, as well as infections. A tumor in some way is a foreign invader. And of course, virus and bacteria are definitely handled the same way. But as in an allergic response, the inflammatory response can be excessive. And for whatever reason, and people are looking into it, and there's some hypotheses and some data, you get an extraordinarily intense response that it's not unique to the SARS viruses. It also occurs in other viruses. It occurs, for instance, in influenza and other viruses as well. But it was something noted very early in patients who were dying before we understood the total spectrum of possibilities. But it was detected very early in our progressive understanding of the biology of this virus. And it's called cytokine storm. And it suggests the intensity of what was happening. It turns out to be also, as things quiet down on the adult front and so forth, we've noticed that the pediatric population seems to be particularly sensitive. It's not a huge issue. They're not thousands or tens of thousands of kids, but there in almost every country, there is a series of children who are affected by it. And it's very serious because the inflammatory cells are in the blood. They're not limited necessarily to the lung. They can go anywhere. And as a consequence, they can affect the liver, they can affect the kidneys, they affect the heart, affect the brain, and of course the lungs as well. And that's called a multi-system syndrome. So you have an inflammatory multi-system syndrome, and it's excessive inflammation, and it can have a fatal outcome. As far as I know, as of today, and there was just a paper published this morning on online JAMA. I didn't see any evidence that there had been any fatalities reported, but a number of kids, and in Israel as well, I know 3 cases at least, have been hospitalized in ICUs. It's a serious complication of the virus, and in adults, especially elderly adults, it can be a fatal complication. It's not common, but it seems to be one of the things that one always has to think about. Now we have an epidemic, know something about the virus and its epidemiology. What do we do about it? How do we manage it? Well, there are 2 basic considerations, and you hear this talked about all the time, not always in a structured kind of way. People argue about it rather than structuring the importance of it. We talk about testing and treatment. Now, those are the 2 ways we really want to think about managing this. First, why do we want testing? Because I think the crucial element of our control now, today, this day, is still an epidemiologic one. Being aware of the epidemiology of the virus, its transmission, the interaction between different people in populations, and having sensitivity to that, you have to have tools to isolate people who not only are sick, who have had the virus, who may have antibodies to the virus, which means endogenous proteins that can defend the body against the virus for future attacks. But you also want to find out if the people replicating, if they have active virus, and they're going to give it to somebody else. So testing becomes extremely important, and you can do you can do it in several ways. Some of the most common ways is an antibody test for COV2 proteins. Okay, you saw a whole bunch of proteins that are elaborated, that are on the surface of the virus, and they should be detectable in blood tests in serum tests, which is part of the blood. If they're present, then you know that this patient has active viremia. And you can, it's not listed here, you can do a test actually for the antibodies to the coronavirus. That's separate. That's actually extremely useful. And you can actually do viral detection directly using a fancy procedure called PCR, polymerase chain reaction, which allows you to produce millions of copies of any nucleotide sequence, which enables quantification, basically. It's a tool, but it's a very powerful tool. And the other things were sort of indirect ways of testing that they're used publicly and they're cheap. One is taking a person's temperature. You can do it now remotely. There's been a flurry of sales of these remote temperature devices, which probably sat on somebody's shelf for 15 years without having a real need. But now there's explosive sale of that device all over the place. And the device that I mentioned to you, which I think is a very nice way of keeping heads up on the possibility of infection and the possibility then of early intervention. If you detect it early, you can be in a properly supervised environment, which may be very important to prevent complications. And so that's testing, and that's why that's important, because you have handle on the one thing that we may have some control over, and that's the epidemiology. The other thing, or the spread of infection. The other thing, of course, near and dear to all of our hearts is this idea of treatment. What are the treatment options? So we think about this continuously, right? There are antiviral pharmaceuticals. We've seen that with a whole bunch of viruses, with flu viruses in terms of pharmaceutical, AIDS virus, rather than the vaccine, has been effective, and there are many therapeutic agents. Some of them are actually anti-cancer agents, which do double time as antiviral agents, because it's the same idea. Cancer is growing quickly, it's replicating its nucleic acid component, and viruses seem to do the same thing, whether they're DNA viruses or RNA viruses. They all also have this chain of nucleotides, DNA, RNA, and that has to replicate. So drugs that do it for cancer may also do it for viral replication. And you'll see that some of the drugs now being trialed are ex or present cancer medications that are being repurposed or used in off-label fashion. And what has to be tested by the FDA? They're already approved, but we don't know that's effective for this, and may have specific complications, because remember, many of the people who it's being tried on are very sick. They're sick in a different way than people with cancer. These people have this pulmonary complication. They may have multi-organ complications. So they're sick in a different way. You have to know that it's safe in your population before the FDA will approve repurposing or off-label utilization. So that and takes time. But that's what everyone is waiting for on these. They know about these viruses. They're thinking about them, and there are a lot of, at this point, there are about 150 drugs, and it may be more than that. I follow it every couple of days, and the figure just continues to mushroom. Some of these are repurposed, and some of these are being de novo synthesized and produced for or in response to this specific epidemic by researchers. Okay, and the only one that is genuinely approved by the FDA and has, according to the Americans, become the standard of care is this remdesivir, which was originally developed to treat hepatitis and colds. And it works by blocking RNA replication in the cell, which we saw was very important, again, because the virus lacks an ability for self-replication. So this, if it can't replicate, it's like a virus on a surface for a couple of hours. It's a useless, non-infectious collection of molecules. Okay, and then there's another one which is highly controversial. It ought not to be, unfortunately. It became a political controversy. But hydroxychloroquine, I have total confidence in the basic safety of this drug. Whether it is effective or not depends. And you have to think, when you think of a disease like this disease, COVID-19, you have to think of it as stages. So you think of it in my stage, and maybe probably all of us, we're worried about prophylaxis. We're worried about preventing infection, which means vaccine in most people's minds. But also there's something called prophylaxis. People have been going to malarial infested endemic sites for years, 50, 60, 70, 80 years. And quinine and chloroquine have been used as a mainstay of prophylaxis for workers, volunteers, sometimes workers in areas with endemic malaria. And malaria is still one of the major scourges all over the world. So these drugs are in use and widely used, prescribed, and people have a lot of experience. And I've seen and talked to people, seen people write about it, their own personal experience as practitioners, and have read about it. And it just seems, if you have no specific counterindication, as you would normally for any drug, aspirin as well, it seems to be a relatively safe drug. Now, whether it's good for prophylaxis, it's being used for prophylaxis all over the world. Whether it's good for early infection before you get significant effects, before you get silent hypoxia, for instance, whether it's useful in advanced disease when you're intubated on oxygen, those are all questions. No two people are the same. You vary as a function of age, a function of intercurrent infection. All these go into the brew, and you mix them around, and you say, this patient, I'm not sure he's comparable to that patient. His brew, his mixture is somewhat difficult. So designing these studies has been difficult, has been a challenge. And usually, if you don't have something like remdesivir, which has a double-blind, controlled random trial, you're forced to use a drug on patients who, by definition, are very sick. They have nothing to lose at this point. They're desperate for some sort of intervention, as is their caring physicians. So I just say just briefly, there are two huge studies. One is well over 1,000 people, and the other one is close to 1,000 people. One from New York, that now is about a month old, from NYU, and the other one is from Paris, in the hands of a world-renowned epidemiologist, virologist, and both of them found from observational studies, which are easy to do in the thick of an epidemic, that it worked excellently. And there are also some anecdotal information that supports that it might be a useful drug. Now, there were two studies that appear to show that it was unsafe and unusable and so forth. Both of those studies, one from Lancet and one from New England Journal, have been withdrawn because they were fraudulent data. So people are rushing to destroy the legitimacy of hydroxychloroquine. For some reason, I'm not quite sure why, because since it's used, it continues to be used for various indications, including rheumatoid arthritis, SLE. People, probably your friends, may be taking this drug. So again, whether it works, it depends on the stage of the disease, how many people you have, and things of that sort. So I wouldn't write it out, considering there's nothing else right now. And there are a few other drugs that are in the pipeline. I mentioned there are 150. I made up this slide, I think about 3 weeks ago, when there were about 30. So things are coming apace, fortunately. And these drugs work on different points of biological vulnerability of the whole cell. The replication of the virus in the cell, of course, is going to be critical. Entry of the virus into the cell, of course, is going to be critical. And of course, a lot of therapeutic efforts are going to be directed at, as you can imagine, that process, especially the spike protein and other membrane proteins. That is the Achilles tendon of the virus, and its need, its obligatory need to be intracellular in a host cell. Okay, now, so that's the story with therapeutics. The bottom line, we're left with one. Vaccines. So let's look at the future with some hope. how do you get a vaccine? Well, there are two immunologic options or possibilities to develop a vaccine. You think immunologically, you can either have a passive vaccine, which means that you already have some antibodies from someone, like it's called convalescent serum. They've been using the serum. It's part of the blood. You fractionate it and you get this yellowish material, take out all the cells. That's serum. And convalescent serum means that some of the proteins in that fraction are antivirus. Okay, we know that they're antivirus based on analytic criteria, not necessarily clinical criteria, but analytic criteria. So this is passive. We can give you for time being, and this is the way hepatitis before the vaccines was treated with immune globulin. That's the way you would be treated. If you were a health worker seriously at risk to contract hepatitis, you would be given this very non-specific therapy. Now, that's called passive immunization, and based on convalescent serum. It has some downsides, of course. This is somebody else's proteins. Now, the body objects to having somebody else's proteins floating around your system. And sometimes, not every time, but sometimes you develop actually an immune response against this immune agent. It's called serum sickness, and it can have very negative effects on the person receiving the convalescent serum. So that's always a concern. You have to watch it very, very closely. And of course, since it is somebody else's antibodies, it's protein, it can be metabolized. So it's passive and it doesn't stay around very long. It's short term. But in the meantime, before a very seriously ill person, to get them over this hump, it might work fine. But that's its limitation. It's not permanent. It's very short term fundamentally. The other side is active. That's the ideal. That's what everyone is talking when they talk about vaccination. That's really what they mean. Okay, and the passive convalescent serum is just in the meantime. It's to a gap filling measure kind of thing. Okay, active immunization is the gold standard, and we want to induce intrinsic immunization against the virus. Now, in all cases, though, what we need to think about very seriously are basic questions. Whatever we give to anyone, whether it's a therapeutic, whether it's a vaccine, we want to know that it's safe, right? And the other thing with respect, particularly to virus, is there rapid clearance of virus? You have to have people who are actively viremic, who have an active viral load circulating in their bloodstream, to know that. Now, because a vaccine rapidly clears the virus, doesn't mean it's biologically going to be effective, therapeutically effective. It has to do one more thing. It has to satisfy one more criterion. It has to be protective. So you have to be able to challenge the person who's been vaccinated to see, in fact, whether he is protected. Now, ethically, you're going to have a problem doing that. So it usually comes up in spontaneous situations. If there are a sufficient number of infected people in the population that you're part of, you'll be exposed, and you'll be able to tell, yes, oh, I went to that place, that stadium, that shul, whatever it turns out to be, and it turned out that there were 50,000 people who were infected. For sure, I've been exposed, and I'm not showing any positive effects whatsoever. The vaccine you gave me must have been protective. Now, doing these kinds of studies at this point becomes somewhat difficult, since we now have, as you saw from the Israeli data at microscopic views, we now have pretty much control of the number of infected cases. So it's very low. So it's very difficult to find large populations where you can find out and determine and explore the protective effect of putative vaccines. Now, there's a lot of excitement, and you hear this in the news all the time about different approaches and new things being done and so forth. There's some old things and new things. They're trying some. I don't think anyone is trying this right now, but there is an approach of live attenuated viral vaccine. Now it's live. It's a real vaccine. It in the proper circumstances it can actually reproduce in a cell, generally cell culture, for instance, or in a person. But it doesn't cause a disease. It's non-pathogenic. So that's what we mean by live attenuated viral vaccine. And that was the approach actually not too long ago, using measles, mumps, rubella, all the childhood diseases, and it generates long-term active immunity, generating an antibody in response to the reoccurrence of exposure to the virus. So it works. It's a real meaningful vaccination. And it's come up recently when people in some communities have resisted being vaccinated for measles, and they've started measles outbreaks as a consequence of that. So it works, it's effective, it produces long-term immunity, and it protects the person and the population. And Rav Asher Weiss has written definitive tshuva on it, that it is absolutely obligatory to immunize your children for measles under that circumstances. The other way, which is actually being used, is inactive viral vaccine. You take a virus and you have tricks to inactivate it, so it's not actually infective. This was used for flu, hepatitis A, and rabies. And again, the bottom line is you get an antibody, and the antibody can be evoked as needed in response to contact with renewed contact with the virus, and protect the individual and thereby the population. You can use protein vaccines. So you either isolate from the virus a protein, or you generate it synthetically. And we have in vitro tricks with the presentation of a protein. We can get cells and culture to make a monoclonal antibody, an antibody that's highly specific to a particular protein. So that can be done in a laboratory, and it's actually done all the time, but not at a level to make it a commercially effective vaccine. It's used nowadays, by and large, as a research tool. And what's really a shining star in the horizon, we'll talk about in a second, is the mRNA vaccine, which is in advanced clinical trials. It's extremely clever. It's an mRNA vaccine. Remember what mRNA is. It's this template that enables the cells, the host cells, to make proteins, right? The virus convinces the host cell to make its own proteins. And so that's what this does. That you put in to the individual receiving the vaccine a little bit of mRNA, some nucleotides, a sequence that provides enough information for the person receiving the vaccine to make a protein himself. And he makes that protein, and the protein circulates in his blood. And the inflammatory cells see the protein, and they say, hey, this protein doesn't belong. We're going to make an antibody to it and neutralize it. And that's what happens. And you get neutralizing antibodies to this protein, which is the same protein that you would see if you were infected in nature. Okay, and this is the concept of an mRNA vaccine. And this is something that the Chinese are using. It's somewhat of an old-fashioned idea. A foreign virus, it's called a vector. Now, a different kind, an adenovirus, rhinovirus, a common cold virus, not very pathogenic, or not pathogenic at all. You take a foreign virus, it serves as a vector that it enables transport from the outside into the genome of a human cell. Okay, and where is it transporting into the recipient of the vaccine's human cell? Information to encode the viral proteins, and in certain cells, these are antigen presenting cells, so they have the capability of triggering the mechanism, the cascade that results in antibodies to these specific proteins. And this is a general schema of how a vaccine is developed. So you start off with design, the concept. You do animal lab tests. You get a cadre of mice volunteers, and you do a series of tests on that. That's called preclinical testing. Then you go to clinical testing, which obviously involves humans, and it's generally described in phases, phase 1 and 2, the initial phases. You know, the safety, you worry about toxicity, and phase 3 is efficacy, and compared to some sort of standards, you may have placebos in phase 3 and so forth, where you have progressively larger number of people, and at all times we're interested not only in efficacy but safety as well. Then you have this big hurdle to overcome. How do you go from this kind of thing, which is a local laboratory exercise, to this kind of thing where you could require millions and millions of doses of vaccine? It's not a small issue. It has to be by scalability. So this is just a breakdown. That was a soft way of presenting it. This is the hard reality of the different steps we have to go through. And the clinical trials, this is just their 5 different phases, aside from the preclinical phases involving our friends here, murine volunteers. This was for the MERS virus. This is just phase 1. Phase 1, phase 2, there's phase 0, 1, 2, 3, and 4. Okay, and at this phase, you're looking at the effects, the toxicity, a small number of people of different doses, right? And so you get really small number of people, and a number of them drop down for various reasons. And at the bottom, you just get a few people, 5 included analysis, 8, and so forth. So this is really a trial, getting your muscles warmed up and so forth for the big day, where you go to advanced phase analysis. Now, what's the story on the market? There are a number of candidates. There are a huge number of possibilities. Again, I think the number is close to 150. I have a slide that I didn't use that had something like 53. The slide is probably a month old. It's now well over 150. And one of the major contenders is this Moderna in collaboration with NIAID, the National Institutes. It's an mRNA virus, and that's basically what I was talking about. It's in phase 2, which is serious clinical trials. There's another one which is in phase 1 and 2. The University of Oxford and AstraZeneca is very interesting because Oxford is the brains behind the vaccine, and AstraZeneca is a company capable of scaling production that Oxford envisions into something that can be useful worldwide as an epidemic solution, as a real epidemic vaccine. So this is a very nice example of a partnership between academia and commercial interests to make worldwide vaccination possible. Okay, and they're fairly advanced. They're in phase 2b3, which is fairly late. So they're on the cusp of actually be able to release a virus. It has been alleged that the Chinese have been aggressively trying to hack into their system and try to unearth some secrets of vaccine production by this team. So far, it's as far as they know, they're aware of it. I've heard reports there's awareness of the attacks, but none of it so far, as far as they know, has been successful. But it's a tough business. And then there are a few entries from Wuhan and the neighboring. This may be also a Chinese, another Chinese. They use inactivated virus, which I haven't seen in many years, and surely doesn't fit in with some of these modern trends. But anyway, what's happening in Israel? I checked a couple of days ago. There were 6 major contenders. There are a lot more that I understand, but these were the 3. There were 6 major contenders in the field that I overviewed, and 2 of the major contenders seem to be this MIGVAX which is a local product. That is, I think, finishing preclinical evaluation. And then the Israeli Institute of Biological Research, which used to be a biological warfare institute a number of years ago, Ness Ziona, but now they also have a candidate which is in advanced clinical trials. This was preclinically trialed in birds, I think in chickens, and the preclinical trials were in mice. But the bottom line, still, we have remdesivir. We don't have a vaccine, and we have one therapeutic agent, which has limitations, can be used in seriously ill people who are on ventilators, and it will reduce your hospitalization stay by 30%. Now that is not a lot to get excited about. Maybe it is. The people who designed these studies were very excited about it, become the standard of care. I hope that there's something to handle people earlier on in the process. For the time being, that's what we have. So this is my slide. This is a semi-medicine, philosophic, asking, what do we do? How do we think about this? What's the way forward? Okay, so in general, and in medicine in particular, we always, when we see an issue and we have to do something, sometimes we have to do some very radical things, and we have to discuss realistically with the person, the family, and so forth, the risks and benefits. You know, are you really going to be helped with this, or is this going to be a personal tragedy? And I've sat in on committees as one of the consultants on patients on how do you decide that? How do you develop criteria? You know, when is enough for a specific patient in a particular setting? So this is real. It's a real-time issue. The risk-benefit. That's what you have to think about. So these are my ideas of what a risk would be in the current situation. It's a sum, non-quantitative or maybe semi-quantitative, of medical factors, the likelihood of exposure, the dose of exposure, and the therapeutics available. So medical factors, we all know. Some of them are intercurrent, medical conditions, hypertension, diabetes, age for sure. Where's my pointer? This is a dramatic difference if you were 39 years old versus being over 80. There's a 100-fold difference in vulnerability there. That's pretty sobering. A likelihood of exposure. Now, we're perhaps in the middle of a second peak, so that's really something to think about. I think it reduces to two factors. One is, what is the prevalence of infected individuals in the population? And two, are they geographically isolated? Are they localized in a particular place? Are you likely to circulate among that population? So if the prevalence is high, the significance to the whole entire population of infected individuals, the geographic distribution may not be as significant. But as in Israel, because of the lockdown, as the general incidence of the virus or infected people was reduced dramatically, there remain conspicuously certain pockets like Bnei Brak and Beitar, I think, certain places like that. And there's a Bedouin community also that now in this recrudescence of the virus represent hotspots of infected individuals. So it has a specific geographic distribution, and it's not generalized in the population. So the likelihood of exposure still for us in this neighborhood, in this environment, for the foreseeable future remains low. We have to watch statistics very closely, but that's how I see the situation now. And we hope and pray on a daily basis. I think over the summer, a tremendous number of the now in serious clinical trials therapeutic agents will come to fruition, probably not before, I didn't mention this before, not before the end of the year is it likely that we'll see a vaccine. A vaccine usually takes 3 years with a lot of pressure. It perhaps it perhaps can be done in 2 years. To do it in the time frames that people are talking about is unprecedented. And it's only because we are utilizing unprecedented technological approaches to this issue that we even imagine that something like that may be possible. You have to consider before you can ethically release a vaccine, the toxicity of which has been tested in maybe 100, 200 people to a population of millions, the ethical responsibility for that is tremendous. So you want to do it in a graded series. You want to do it long enough to know what are the idiosyncratic responses to this virus. Is it really safe? And that kind of time frame of 3 months, of a year even, is really very difficult to envision that you're going to get that kind of information back. But that's the hope that there'll be hard enough data and the regulatory red tape will be cut through chick-chock. And we will actually have an ability to move from phase to phase and accumulate enough data and enough certainty of the safety issues that we can go ahead and take a chance of its efficacy. The efficacy will be the last thing that we'll be able to have in control, but we want to make sure before we go forward that we at least are secure that what we're allowing to be used in a general population of millions of people throughout the world is safe. Now, what to do before the therapies? Remember, there's still only one. No virus, no vaccines. So these are all practical things you hear all the time. The bottom line is not getting affected, and 2, not infecting others, right? And you start off with social distancing. Some examples, we all know what that means. I think that that's extremely important. Um, there's a question about whether it really is transmissible by aerosols, exactly how that could be. And there are many arguments, both technical at the engineering level, at the medical level. It's a very complicated issue. I could speak about that for an hour all by itself. What is the mechanism of transmission and infection in this particular virus? That again is another aspect of a novel coronavirus. But social distancing at least answers that question, whether it's a real question or a real concern. We don't know at this time, but it's realistic at everything we know, and it seems to work. Social distancing lockdown dramatically reduced the number of infected cases. We saw the data altogether. So it works for whatever reason. Wear masks. It depends on the setting, what kind of masks you wear. There are situations where I'm at home, I don't wear a mask. I go out to walk the dog. I wear my mask. When I go to the open minion, I wear a face mask. I'm going to go in about 2 weeks, first time, in about 3 months to a concert, and I'm going to wear 2 masks, just like the medical teams do with a higher level of exposure. Why am I doing this? Because the first 2 or 3 cases in America that were clearly indigenous cases were in a church. And they were able to trace I think it was 2 people developed the disease from a person in the row behind who had been exposed by some way that they could track also. So the sitting in a large auditorium, even with some degree of spacing, puts you at risk for a higher dose and exposure than you might normally. And I would take a little bit more care and have concern for those issues. I carry a bottle of disinfectant on me. I go walking uh, probably 5 or 6 kilometers a day. I go for a far walk up to the top uh, near you guys. I live at the bottom of the hill below the embassy, so it's quite a walk. And I do touch things on the way, and periodically I will take out my little disinfectant, not just washing your hands, but actually the disinfectant. It's a small container that fits into my pocket, and I'll do that periodically. Um, I don't use gloves because of that. I've disciplined myself to wash my hands frequently. If you're in a situation where you think you'll be touching something, touching your face and whatever, then perhaps that's good. But the gloves won't help against that. So I would say that I don't think the absorption from the skin is significant. So if that's what the glove is protecting you against, uh, I don't think it's necessary from that point of view. If it's to protect your face, if you have your glove on, I don't see how that helps. So gloves are still recommended. And if I was working with sick patients, that's probably what I would wear because that's is what is suggested. Although it's not quite clear that is really more efficacious than frequent disinfection. When you're in the throes of an emergency room dealing with tens of people who are potentially infected, that may not be a feasible solution to wash your hands every 5 minutes or so forth. So gloves there does make some sense, does have a rationale. Now, very important, based on what I said before, don't touch your face, eyes, nose, and mouth. Okay, these nerves which innervate your mouth, your tongue, your nose, and your eyes, all can transmit a viral particle backwards, and perhaps lead to general systemic viremia. In other words, from that source. So perhaps it is somewhat superstitious behavior. It really fits in with some aspect of we know about the neurobiology of central nervous system infections. So it's sensible, although it again, one of the many things for which there is adequate data to know whether it's really true and necessary. And the same thing with disinfecting surfaces, cleaning surfaces. Um, the CDC has changed its recommendations on its website in the last 2 months, 3, 4, 5 times. I can't keep track of it. I read one time that they specifically said that after saying that it can survive for 17 days, that they reevaluated that and they found that on surfaces the virus dies very quickly and disintegrates into its various component molecules and is non-infectious. That was recently, probably about 2, 3 weeks ago. So I have stopped disinfecting things brought into the house. I disinfect my hands frequently, but I don't go the full mile in that regard. Now, the washing your hands versus disinfection, that's a reasonable thing to ask. Disinfection will kill infectious agents. Uh, there was this brouhaha about using bleach and how what Trump said was interpreted. Any event, whatever, bleach kills the virus within minutes very clearly. A good disinfection. Not all disinfectants are approved by the FDA. Some of them are clearly fraudulent. I have no idea whether the ones that are supplied in Israel are fraudulent or not. If they contain isopropyl alcohol, that is accepted as significant disinfection. And I could rely on something like that. Other things which don't indicate the proportions of the various components, you don't know whether they have isopropyl alcohol or something else. It's not clear. I would avoid them. They're probably imported from China, and honestly, at this point, I wouldn't believe anything that the Chinese said. I can go on on that subject for quite a while, but I won't. So I'd like to leave talking about some disturbing things, perhaps some hopeful things. And we're in a tight spot at this particular moment. And I would say, taking the advice or the parallel from many Gemaras that always ends in a positive note. Even Moed Katan, which discusses in the last chapter the halachot and dinim of avelut, it ends with a nice story. And with that in mind, I wanted to say, let's make the most of your day. Life at home can be nice too. Now I'll take questions.
If anybody has a question, you have to unmute.
Don,
Yes.
this is Jonathan Porath. What's the category of Putzot Sikun? What do you think of the efficacy of going into shul for davening as opposed to video or outside or something else? What are your thoughts on that, please?
It's very difficult to know, given those four things I outlined as risk factors. You have to make a number of assumptions. You have to start with your own basic status as the first known, and the second thing is what is the likelihood that there's going to be infection in this population? Is probably extremely low. So that if you social distance, I would imagine that that's a reasonably safe thing to do. But I found watching people behave as they come and go from shul, even if they sit in shul and they observe the proper spacing for social distancing, as they leave, they can't help overcome the instinct to come close to somebody that whose company you've enjoyed for years, and speak to them at that kind of distance. I find you people naturally, just watching people come out of shul, what happens? They have a magnetic attraction to come together to talk the way they normally did. Whether I come from a nation of prophets, a long line of prophets. I don't claim to have any degree of that skill at all. So I don't know, in fact, that there's no one that you're going to contact in our general population that is not actively infected, maybe from a grandchild or something like that. There are a number of different routes. I assume all of us have not been infected. So it means that if infection is available in the population, we are potentially at risk.
Don, this is Bob Rosenshine. Do you have grandchildren?
Yes. What did I do about it?
What's the right way to see them?
Okay. The way we did up till and more or less continuously is we saw them outside. In other words, we have a long closed door with doors opening. It's very well ventilated, and we'd sit on one side, and they would sit 3 meters or so away. The 2 meters, and of course, everyone wore masks. That is needless to say. There's all sorts of new data emerging about just talking. If droplet infection is truly the method, the source, the vector for transmitting the virus, then those droplets are transmitted by just talking for at least 3 meters. So 2 meters is a minimal amount. So we stay about 3 meters away. We're all wearing masks, and it's well ventilated.
You don't have meals with the grandchildren?
I don't have meals with the grandchildren.
Not even outside.
I have a question. Has there been a change about the way ventilators have been used, or when ventilators need to be begun to use? to be used because of all the questions about whether people do worse when they're put on ventilators and physicians and so forth.
Okay, that is a very complicated technical issue, and I won't address it because it really involves a number of very complex clinical issues as well as technical issues. It's a kind of question that a intensive care respiratory specialist has specific guidelines that are evolving almost on a daily basis. So it's a highly changeable and changing situation, and it's very complicated.
Don,
Yes.
Laurie Blandis.
Let me just backtrack. Do I have meals with my grandchildren outside? I caught that at the very end. The answer to that question, I've started to go to like parks entirely open and sit at a distance, and I would picnic with people. At this stage, I'm confident that given my my 4-point risk equation, that the incidence in the environment is sufficiently low in that setting that I'm not concerned. I wear a mask. They're at a distance, and if occasionally they move back and forth within a domain that might be a risk domain less than 2 meters, the fact that it's outside and there's flow and there are not a lot of people around, I think that works very. I've done that a number of times, and I feel totally comfortable doing it that way.
You can't eat a meal. By the way, I think you can stop sharing the screen. You can't eat a meal with a mask on.
That's true. So you have to appropriately distance and figure out what you're going to do about that.
Yeah. Don, could I ask you to stop sharing the screen and then we'll see you better?
Okay. Just a second. I'm
Don?
not get. Oh yeah, I see it. Okay, fine.
Yeah.
There it is. Okay.
Don, my
Yes.
name's Tamara. I've become concerned because recently we did have an outbreak in this neighborhood, given that a lot of the schools that were shut down, that there was exposure, are actually a lot of children from our neighborhood specifically. So how does that change the equation?
I would say if there is a reasonable chance that you could come in contact with somebody who's been in contact with that person who has not been infected. Like there are families who've had
Mhm.
2 children who are likely have been infected. They've had mild fevers. They were in a situation where they were in the setting of an infected person, a teacher or something like that in
Mhm.
school. I have 2 grandchildren in that situation. The likelihood is everybody in the family is now immunized, spontaneously immunized. So that's what you have to decide. Are the people that you are contacting from that family likely to be, there is no easy way of knowing without testing. I mentioned that the testing in managing the epidemic was critical. Unfortunately, there isn't a test we can go to the local drugstore and buy, which would facilitate that. You would know whether, in fact, you have already generated antibodies to the virus. That'd be very helpful, or whether you still are at risk. And everybody in the neighborhood. The government is not willing to do something like that, and there is no private initiative that's generating, as it was Abbott in America, that's generating tests for us to answer that question. Without an ability to test vulnerability of our local populations, there's no way of definitively asking the question. You can be, again, you have to figure in your own personal medical status. Everything else being the same, there's perhaps a low risk overall of you actually contacting somebody that has active virus to transmit. And even though there isn't a vaccine available, and you're in relatively good shape, you could take a chance. It would be so much better to know that the environment, in fact, doesn't contain anyone at risk that you're circulating. But right now, we don't know that.
Yeah.
That's the importance of testing.
Can I ask a question?
Don,
Yes.
Laurie Blandis. Terrific lecture, by the way. Thank you.
Thank you.
This second spike
Yes.
is different to the first one. Predominantly younger patients, and so far a very, very low mortality rate, and I think severe cases. As an advisor to the government, how should we respond as a country to this second spike?
The answer is we've learned some things from our initial exposure and experience, that you want to continue to isolate people at high risk. That's good advice, and it remains good advice. Younger children seem to do better. We don't understand the mechanistic considerations for cytokine storm, which does seem to affect some children. But by and large, children have very mild. It was alleged in the original epidemic. The original epidemic was very complicated because we were scrambling around trying to find our bearings. We're trying to define what is important. We now know what's important. We can social distancing, appropriate separation of high-risk populations can actually save them from contributing to the pool of infected individuals. That works. That will continue to work. So, in answer to Tamara's question, it's a parallel case. If we have a general idea of what the incidence of infected people in your population, in your neighborhood is, we can act appropriately. If we don't, then we really can't. And what the government is doing by closing the schools seems appropriate, and putting everybody that's been in contact with the children that attended the schools in isolation is appropriate. That's what we learned was effective the last time. Grandparents of those children by this time should be sufficiently aware that for 2 weeks those children are in isolation and you have no social contact with them. So we've learned some things from the past. Most of the things we have learned are epidemiologic. We've learned to police and handle ourselves with respect to the potential for viral exposure and infection. And that remains the key lesson from the past, which is now relevant, highly relevant to the present. The other factor was in terms of dose. Again, it's the geographical concentration. I think 1 or 2 people in a neighborhood probably do not represent a tremendously high dose. They wouldn't be considered a hotspot. In areas where we have, if you had a small, a block or 2 or 3, where you had most of the kids from a school, and you had a significant number of them that were infected, or 2 or 3 teachers that were infected, that would be a different situation. And in fact, if I lived in a neighborhood like that, I might even think of leaving for a while and maybe staying with my children or something like that in an appropriately arranged quarters within their house. But barring that sort of extreme situation, I think what we learned from the past is how to manage ourselves in relationship to sources of infection.
Don, this is Sam.
Do we need a lockdown again?
So far, we don't. I think we're handling it appropriately. We're taking the geographical approach, and we're focusing on areas, which is the way we did it initially. That was so successful. Israel is one of the most successful countries in the world in handling the corona pandemic, because we have this somewhat draconic approach to isolation and identification of infected people and keeping people at risk away from sources of infection.
I just wanted to ask, I've read that the immunity of those people who've had the disease is very short-lived, and that therefore the idea of a herd immunity is unlikely to work.
Well, herd immunity has been misused by the press. It's not a spontaneously arising immunity in a population. Herd immunity occurs when the vast majority of people in a population have been vaccinated. And this was a factor, for instance, in the polio epidemics, where there is oral fecal transmission. And we know that once you have a highly immunized population, the significance of that in terms of generating a general herd immunity works well. But to the press was presenting the idea as if you have enough spontaneously infected individuals, you would naturally develop herd immunity. I haven't heard any serious epidemiologists endorse that approach. And in fact, from a medical point of view, it seems somewhat reckless and dangerous.
The spontaneous immunity, is it long lasting? We don't know that yet, or what?
Just a minute, I have a slide. Let's see, I found this from May 19th from one of the sources that I check daily. It says, new data suggests people aren't getting reinfected with the coronavirus. This is May 19th. People who recover from COVID-19 but later test positive again for the coronavirus don't carry infectious virus, a study finds. So the virus has been effectively neutralized.
If you have hypertension, they say that you are more at risk, even if it's controlled. So if you're on medication, and your hypertension is under control, would you still be more at risk?
I've thought of that an awful lot because I happen to be in that age group and share these comorbidities. The way it seems to me, I may be proven wrong, but so far I haven't seen any data, and I've been looking. It seems to me that we're looking at two different levels. We're looking sort of microscopically at the medical status of individuals like you and I, and they're looking globally at population risks, like in Italy, in the United States, and looking at the number of people in that age group who are vulnerable. And if you look at that population in that, I mean, vulnerable in terms of potential mortality. And if you look at that population, what are the kinds of intercurrent diseases they're going to have? Well, people over 70, over 65 are going to have hypertension. There's a very high incidence of hypothyroidism. There's probably a very high incidence of type 2 diabetes, especially in Jewish populations. So there are all these intercurrent diseases that come pari passu with the aging process. So I think when you say that people over 70, over 65 are at increased risk, I think that's true. And whether this is an epiphenomenon, which means it runs parallel with it, but it's not causal, or whether it's causal, that means hypertension, diabetes, whether it's causal or not, I can't say. Now, that's one side of the answer. The other side, I see no relationship between hypertension. I can talk about ACE2 receptor inhibitors in a second, but I see no relation between hypertension and any level of vulnerability that I can imagine. Maybe it's true and someone will have to show it, but so far I haven't seen it. Now, diabetes is different. Diabetes produces low level of immune deficiency. I have, in my career, seen some bizarre infections attributed to, in the setting of someone with out of control. I mean, not seriously out of control, not in hyperglycemic coma or something like that, but an ordinary person running his life with diabetes, not taking his insulin very regularly, eating as much ice cream as he wants to, basically avoiding confronting, which is a normal human reaction, confronting the fact that he has a potentially fatal disease like diabetes. So if you're well controlled, that's very important. A lot of the basic defects, the enzymologic defects, immunologic defects in diabetes are better. The metrics are better in patients who are well medicated. So of the various things that are endemic to people over 65, over 70 for sure, characterize in our modern Western civilization people who are frequent sufferers from these various diseases, and except for diabetes, I don't see any causal relationship. I think it's by and large an epiphenomenon of the fact that this is the age of vulnerability. Now, that being said, it is absolutely the case in almost every parameter. Early in my career, with NIH, I was part of the Baltimore longitudinal study. I participated in a study that watched people age using quantitative parameters of aging. And what we found, this was a period where the popular young culture viewed anyone over 30 as being decadent. You may recall that period. Well, in fact, that's what our data showed, that almost any parameter, whether it's a hormonal parameter, whether it's a neurologic parameter, endocrine parameter, psychological parameter, neurological parameter, they all go up to about age 30. They progress in a good way. And at age 30, they begin to break, and the curve begins to descend for the rest of your life. So at some point, probably between 65 and 70, you reach a point, there isn't a dramatic change in the curve. The curve looks, the slope is about the same. It's not that it suddenly falls off. That's not what happens. It continues gradually to decay. But everything is a matter of, sort of use the metaphor of risk benefit. You get to a level where the body cannot maintain enough basic function to counter some of the basic challenges that you may be encountering. So that's a threshold. 65, 70 is a threshold where you begin to see various things presenting that way. And the immune system functions exactly the same way. Your immune system gradually is deteriorating in capabilities. So people, I've seen people age of 90 go into a hospital with a minor infection, maybe a minor pneumonia, and the course, the terminal course, is a matter of days. It's shockingly rapid. It's because the threshold for those sorts of events has dropped dramatically. Your capability to defend yourself at that age drops dramatically. So that people over 65, it's because you're on a decaying curve where functionality is dropping over years, and you reach a point where your vulnerability to infectious agent increases. So I would say automatically over 65, if not everything else aside, your ability to defend yourself against infectious agents is low. Diabetes may contribute, other things may contribute. Certain things I don't see how they contribute whatsoever, and I believe they're entirely epiphenomenal. Okay.
Thank you very much indeed.
Thank you.
Thank
Okay.
you very much. Excellent.
My pleasure.
Thank you very much.
Thank you
Thank you so much.
very much.
Good night, everyone. Thank you.
Don.
Thank you very much.
Thank you.
Thank you very
Bye.
much.
Thank you. Thank you, Don.
Safe and well.
I'm ending the meeting.
Very nice.
Excellent presentation.
Glad you enjoyed it, and hope you learned something.
A lot.
Good. That's important. That's what you gain. I'm a
Thank
simple
you.
Malaman.
Thank you.
Bye-bye.
Bye.